Thanks for the introduction. I'm Chaoyue, and today I'm going to talk about our work on quantitative susceptibility mapping in UK Biobank brain imaging. I have nothing to disclose. So I'll start off with an overview of the UK Biobank imaging study, which is the foundation of this presentation. So UK Biobank imaging study is aiming to scan 100,000 subjects with a multi-organ protocol. Currently, data from 60,000 subjects are available. These subjects are largely healthy. These subjects have undergone extensive baseline measurements, including environment, lifestyle, and genetics, and in the larger UK Biobank project. So these subjects are largely healthy when scanned, but they may develop a range of health issues over the coming years. So for that reason, a particularly valuable feature of Biobank is that we have long-term access to subject health records. As shown on the right-hand side, multiple modalities were included in the brain imaging protocol. So my work has been focusing on the susceptibility-weighted MRI, in particular to do quantitative susceptibility mapping, or QSM, with this data, which is what I will present in this presentation. So how do we get a sense of the scale of these numbers of subjects that will be scanned in UK Biobank? Here I will use a video to give you a visualization of the size comparison to typical imaging studies. So a typical imaging study commonly has a few hundred subjects. If we have a few thousand, that's considered to be big. But here comes the UK Biobank cohort with the ultimate 100,000 subjects. Given the age range of UK Biobank subjects, in a few years after the imaging scan, many subjects might develop a range of neurological disorders, including Alzheimer's and other diseases. So in addition, UK Biobank is powerful as a result of the combination of a number of subjects and the breadth of linked data. As shown here, subjects have been extensively characterized and consent to allow access to their long-term health records. So with Biobank, we could learn the relationships between various biological measures and health outcomes to enable a prediction of their long-term health outcomes. So this is a quick overview of the Biobank imaging study. Next, I will give you a brief overview of the susceptibility MRI protocol in UK Biobank. So as UK Biobank is aiming to scan 100,000 subjects that require high throughput, which leads to a limited scan time. So for the SWMRI acquisition, it only takes 2.5 minutes. So they were only able to get 2 echoes and a very thick slices, 3 millimeter. So which is not ideal for T2 star mapping or QSM. But nevertheless, Biobank saved quite uncombined data for both magnitude and phase images on the right-hand side, which is great for QSM. So magnitude data have been processed for T2 star mapping. So my job is to process the phase data for QSM. So in the first presentation, Professor Munger has given a great overview of the QSM techniques, but just in case some of you just joined us, so on the right-hand side is a typical brain QSM images. In essence, the image contrast of QSM is driven by tissue constituents with shifted magnetic susceptibility. For example, iron content in subcortical regions indicated using the orange arrow here. So iron is paramagnetic, so it is positive, appears bright on QSM images. Myelin, on the other hand, in white matter, is diamagnetic, so it is negative, highlighted using the blue arrows, and it appears dark on QSM images. So this is kind of the contrast of QSM, and this QSM is a quantitative map. So to do QSM, a multi-step processing of the phase data is needed. Here I'm showing the QSM processing pipeline we have developed for the UK Biobank data. You don't need to read this pipeline. I just want to highlight that there are multiple algorithms available for each processing step, indicated by the blue arrows here. So we have performed extensive evaluations of these algorithms and selected the optimal combination of them for this specific data set. So this pipeline was used to process data from the recent release of 60,000 subjects. And given it produces voxel-wise maps, so you can imagine that QSM data in UK Biobank is huge. So to do analysis, we have to reduce the amount of data that we need to work with. So in UK Biobank, we commonly produce summary measures that are a set of scalar values extracted from each subject's imaging data. So these summary measures are called imaging-derived phenotype, or IDP. So here, I will focus on IDPs targeting the subcortical regions from the QSM data. So we calculated the median susceptibility in 8 subcortical regions, separated by left and right hemispheres. So by doing this, we reduce data size to a few numbers per subject, which makes it possible to perform analysis computational-wise, so such as associations with genetic data and phenotypic data. So next, I will go into show some example results from genetic associations. So in UK Biobank, they have collected genetic data. So the genetic data collected consisting of over 20 million genetic variants per subject. A genetic variant is a site in DNA that varies across subjects, and we also have QSM IDPs from the same set of subjects, and also have variants across the population, so we can study the cross-subject correlation between them. And we've done this correlation analysis for the whole genome, which is called a genome-wide association study, or GWAS. So here I'm showing the GWAS result for a single IDP median susceptibility in right thalamus using what is called a Manhattan plot. So where each point represents one genetic variant, and x-axis gives its location on the genome, and the y-axis gives a significance of its correlation. So 2 horizontal gray lines indicate a standard significance threshold for a single phenotype GWAS, and after Bonferroni correction for the significance threshold. So any genetic variants that pass threshold or color-coded in black, we see that significant associations are clustered together, such as here in chromosome 4. This is expected due to an effect called linkage disequilibrium, or LD. Here we'll only focus on the top variant of each LD, which is called peak association. So these genetic variants have their own unique IDs, as you can see here. So then we can use this ID to look for related genes. And in this study, we identify genes related to a broad range of biological functions, color-coded in different colors, including iron, calcium homeostasis, myelin or glia, and brain development, and general regulations, including regulations of gene expressions or functions. So this is just for a single IDP. When we combine our 6 or 16 QSM IDPs, in total we identify 292 peak associations. So here I'm showing you the summary plot for QSM GWAS with all ROIs, where the y-axis represents each ROI, and the x-axis gives number of genes associated with a given ROI, and different color means the different biological function of a given gene. And as you can see here, iron related genes are in red are associated with every ROI, and for ROI here, substantia nigra, globus pallidus, putamen, and caudate have the largest amount of genes correlated. So and these four regions also have the highest amount of iron among all these ROIs, and they are one of the highest amount of iron across the whole brain. So this is IDP level results. What we can also do is that we can do this at voxel-wise level. So because producing IDPs is an efficient way to reduce data size and to discover new associations, but it loses the rich spatial information in our MRI voxel-wise maps. So after identifying genetic variants of interest, we can further explore the spatial patterns of these associations. So once these QSM maps are aligned to a common coordinate system, for instance, MNI space, we can correlate each genetic variant of interest with susceptibility values at each voxel location to perform voxel-wise correlations. So here I'm showing the correlation R-value map for variant related to gene HFE, which is related to iron absorption. R values are plotted as a color overlay here on top of group averaged QSM maps in MI space. So yellow-red indicates positive R and blue indicates negative R. So we can see the associations are mostly in subcortical regions, including putamen, subregions of thalamus and substantia nigra, but also in regions that are not captured by our ROI, for instance, the red nucleus and regions in the cerebellum, for instance, the dentate nucleus. So, and most of these associations are positive. If we look at another variant, genetic variant related to a gene that is related to iron export, the first thing to notice is that most of the associations now are negative, and the spatial pattern is quite different compared to the top one. And for this variant, it's mostly associated with globus pallidus, not the putamen. So these are a few examples to demonstrate the genetic associations with QSM IDPs. What we can also do is that we can calculate heritability of these IDPs. So heritability means the fraction of IDP variants that are explained by a linear combination of all the genetic variants. So just to give you a sense of what this means, if you look at the IDP that has the highest heritability here, so medium stability in left putamen. So the center circle means the heritability estimate, and we have arrow bars at side. So this heritability around 0.35 equals to half the heritability of human height, which is known to be highly heritable. So in addition to genetic analysis, subjects has also been phenotyped, which offers us the opportunity to explore phenotypic associations with QSM IDPs. So in UK Biobank, the non-imaging phenotypes consist over 17,000 variables. So we have grouped them into 17 categories, including lifestyle, blood assays, health outcomes, and others. So similar to GWAS, so there's variations in these non-imaging phenotypes, and we also have variations in IDPs. Then we can do a cross-subject correlations. So in total, we identified significant association between 251 phenotypes with QSM IDPs. So again, I'm using a Manhattan plot to show the phenotypic association results between a few phenotypic categories and our QSM IDPs. So each circle here represents one phenotype. A different color indicates different IDPs, or in other words, different subcortical regions. The x-axis here now represents, indicates different categories of phenotype that belongs to, separated by the vertical dashed line. And given the large number of associated phenotypes, I only highlight a few associations of interest. As aforementioned, iron is one of the major sources of contrast in QSM, so we identified many genetic variants related to iron homeostasis. So not surprisingly, the phenotype that has the strongest association with IDP, QSM IDPs across all phenotypes is hemoglobin, which is positively correlated. And we also identified associations with alcohol intake frequency, smoking status, and they are all positive correlations with QSM IDPs. And drinking and smoking are known risk factors for a range of diseases. And this Manhattan plot is associations with other three phenotypic categories: cognitive phenotypes, health outcomes, and mental health. As you can see, again, many significant correlations popping up, including diagnosis of anemia, which is negatively correlated, and also diagnosis of diabetes, which is positively correlated with QSM IDPs. So similarly, we can do this voxel-wise correlations with a given phenotype. Given that we are running out of time, so I just want to highlight that you can see the spatial patterns for the strongest association with hemoglobin, again, across the subcortical regions and also regions that were not captured by ROI, for instance, cerebellum. And for diagnosis of anemia, we have opposite signs of the same kind of spatial pattern in terms of associations. And in conclusion, UK Biobank will produce a QSM data resource. We already produced a QSM data resource that is 2 orders of magnitude greater than any existing QSM studies. And we found that susceptibility measures in the brain are associated with many genetic variants, phenotypes, and health outcomes. And our data have been made available to researchers worldwide via UK Biobank, so everyone can use that now. And so it will be a powerful open data resource for many studies, for example, studies on brain iron in aging population. And this work was done in Oxford, so I'd like to thank my mentors and colleagues in Oxford and collaborators in this work, and thank you for your attention.